Okay, well, it's my pleasure to introduce you to Dr. Pamela Laureto. She just recently passed her dissertation defense at Western Michigan University with a grueling committee, I understand.
Aren't they all?
They are. They're all grueling and very nitpicky, and she passed with flying colors, so she is now a full doctor. And she comes to us from Aquinas College, where she was a saint. Isn't that right?
Oh, I guess so, yes.
She was a saint, and now she's going uphill here. So it's been 10 years, actually, since she started this dissertation research program, and she's been with us for 12 years as an assistant professor, associate professor, and I assume full professor now for 10 years. So we've done really, really well to have her here. She's taught botany and Biology 152, which is our diversity course, as well as several other courses here, so she's very well rounded. And today actually, she's going to talk to us about the Allopolyploid origin and population genetic structure of the rare Solidago Houghtonii, or Houghtonii, as she says it. So without further ado, I'm going to turn it over to Pam.
Thanks.
And do you want questions or no?
Well, I think the best way that this is going to work is if we kind of just go through the presentation. It's because of the audience here, you're not my advisors, so you weren't aware of everything I've been working on. I'm going to try at a few places to do a little bit more explanation than I would have done for them. The dissertation was done in sort of two parts, so what I'm planning on doing is going through the first part, and then we'll just sort of take a break, give those of you that this was about as much time as you were planning on to commit to this, an opportunity to leave and go get some food and that. Those of you that want to hear the full thing will reconvene in just a few minutes after people exit. I think that will work best to give people flexibility in how they want to handle that. The second part is actually the population genetic piece, and I think there's some really cool stuff there. So hopefully some of you will stick around and we'll do the full thing. I know the Biodiversity Club that asked me to do this presentation, and I'd like to thank them for asking me to present to you guys. They wanted the full-fledged thing. They did not want the watered-down version for a general audience because, I think with very good reason, they want to see what it might take someday for them to go through a graduate program and what they might end up having to do. So with that, we'll get started. So I'll start here by introducing you a little bit to the plant that I've been working on. This is called Solidago Houghtonii. Most of the time, we'll refer to it by its common name, which is Houghton's goldenrod. It is a member of the Asteraceae, or the aster family. Now, Houghton's goldenrod is a federally threatened species, so it has protected status on that endangered threatened species list. And it has this name because it's named after Douglas Houghton. Douglas Houghton was Michigan's first state geologist, and on one of his geological surveys along the northern shores of Lake Michigan, he discovered this plant that he was not familiar with. So he collected it and sent it off to one of the North America's premier botanists, Asa Gray, who named it after him. And so we see Houghton's name throughout this state, Houghton, Michigan, the lake and that. So that's where it got this name, Solidago Houghtonii. The plant is characterized by having a basal rosette of leaves, and then its inflorescence is said to be corymboid, or in other words, a flat-topped inflorescence. So its appearance is similar to other goldenrods, but with that flat top, easy to identify if you're on these shoreline habitats. Now, Houghton's goldenrod is an allohexaploid species, which means that it is believed to be of hybrid origin. So it has extra sets of chromosomes, a polyploid. So one might expect that a hybrid species would be closely related to its parental species, and that it would share some morphological similarities and characteristics with those. And so Houghton's goldenrod is in the taxonomic section, Tarmacoideae. These three plants are in that same taxonomic section, and similar to Houghton's goldenrod, they all have this basal rosette of more or less linear leaves and a flat-topped inflorescence. This plant here, notice white flowers, not the typical yellow that we see for most goldenrods, but yet it is a goldenrod. Solidago uliginosa has more or less a wand-shaped inflorescence, but it has also been proposed as a potential parent of this hybrid plant. So, how can an allohexaploid form? This scenario was presented in 1979 by John Morton, and its basis is the fact that Solidago tarmacoides and ohioensis, in nature, they form a naturally occurring hybrid species. So this setup does occur in nature, and that hybrid that is formed from them is believed to be sterile individual, as hybrids frequently are. But plants can do many interesting things when it comes to reproduction. And so one thing that they can do is to end up forming diploid gametes. So if we get a diploid gamete, then that plant would be able to either cross back to the yellow flower as Morton presented, again involving a chromosome doubling of that hybrid leading to a hexaploid, having 3 sets of chromosomes. In addition to just a back cross, you could have a third species that's involved in this formation as well. Now, like many rare plants, Houghton's goldenrod has a very narrow geographic distribution. There are about 70 known populations, some of them ranging from as few individuals as maybe 13 to perhaps several hundred individuals. But these are patchily distributed along the shores of the Great Lakes, mainly in Michigan, but a few populations in Ontario, Canada. In addition, there are 2 populations that are disjunct from that primary distribution. One of those is here in Genesee County, New York, and the other disjunct is in Crawford County here in Lower Michigan. Now, Houghton's goldenrod is associated with a geographical feature known as the Niagara Escarpment. The Niagara Escarpment is the wall of an ancient sea, believed to be of Silurian age. And so back at that time, the whole state of Michigan, in fact, this entire area was underwater under a sea. And so the wall of that sea is composed of a dolomite limestone, and it forms all along here and ends up, the end of it is over here near Genesee County, New York. So Houghton's goldenrod is associated with these alkaline habitats of this escarpment. The only population that is not associated with the escarpment is that inland population in Lower Michigan. However, that population seems to have some genetic differences. A single plant, chromosome numbers were looked at for a single plant by a different researcher who believes that he sees an octoploid number of chromosomes in that population. And so one thing that we're looking at then is this actually Houghton's goldenrod or not? If it's an octoploid, a different number of chromosomes, it would likely be then renamed as a different species. So we'll look at that as well as we move on. Now, in addition to Morton's hypothesis of hybrid origin, a different researcher, Jim Pringle, and his ideas are reported in the Federal Recovery Plan for Houghton's goldenrod. And Pringle's hypothesized, based on morphology, that Houghton's goldenrod, or what we call Houghton's goldenrod, is actually 4 separate entities. And so the boxes indicate where he thinks these different entities are. And here we see the plants that he's presenting as the parents for those different entities. So the first aim of my research project here was to assess the origin and evolutionary relationships of Solidago Houghtonii. I wanted to know, is it actually a hybrid species? If it's a hybrid, who are its most likely parents? And then, did this form once, or as Pringle has proposed, has it formed multiple times? And so those are the questions that we're going to try and answer first. And so to do that, I will use both nuclear and chloroplast DNA to answer these questions from a molecular point of view. Now, I'm not going to go into a lot of details on the methods. Those of you that are interested and want to know more about those, write that down, and we'll talk about those during the question period at the end. So I'll highlight just a few things. One thing that does need to be highlighted, though, is how the collections were done. We're going to compare our proposed hybrid here to other closely related species. So I sampled 25 other species of goldenrod, which included all the sympatric taxa, so all the goldenrods that live in and around Michigan and Canada. Also then sampled Houghton's from each of those different geographic entities, and all of the proposed parents. DNA was extracted from a single leaf that was collected from the plant, so non-destructive sampling, right? It's a threatened species. And then that DNA was used in polymerase chain reactions, or what we'll call PCR. Now, I'm going to talk first here about nuclear DNA, what we find from the nucleus. And the regions of DNA that I was using are part of the nuclear ribosomal DNA. And so there are two internal transcribed spacers, ITS1, ITS2, and there is an external transcribed spacer. And so these are transcribed, but they are then cut out prior to, they're thought to help in folding up the ribosome structure. Now, since I want to evaluate the hybrid status of this species, that's going to involve doing molecular cloning. And so I cloned actually a very large region, 3,055 base pair region from sort of the middle of this external transcribed spacer through ITS2. That then allowed me to individually sequence these much smaller regions of DNA, but I could connect them together, what all came from one clone. And so that's how that DNA data was collected. The DNA sequences were then analyzed phylogenetically, and I also analyzed them with a separate method called statistical parsimony network analysis, and we'll see those results here in a moment. So, what can we predict when we're using nuclear DNA? Well, with regard to our first question, is Houghton's of hybrid origin? Here, the nuclear DNA is biparentally inherited. Both mom and dad are contributing to that. And so we would expect that from the direct sequences of the ITS and ETS regions, that we would find in our hybrid what we refer to as polymorphic nucleotide positions, and that these polymorphisms would be additive between the different parental species. And so this is what a polymorphism would look like. Here I have just a very small section, just a few nucleotides. When you do DNA sequencing, you use a fluorescent dye label that then the genetic analyzer will read and produce these colored peaks for you, which correspond to the 4 different nucleotides in DNA. So if parent number 1 has a cytosine at positions 3 and 5, our parent number 2 has a T and an A at position 3 and 5. Then in their hybrid offspring, you would see these polymorphisms. Both of those nucleotides are appearing at that position, and they're additive between the two different parents. The T from parent 2, the C from parent 1, and we see both of those peaks. So when you find that, that's very good evidence that you have a hybrid species. So here's just one polymorphism that was recovered from the ITS region of the nuclear ribosomal DNA. And these are several different individuals of Houghton's goldenrod from these different geographical regions. And we see the two peaks occurring in the same position for all of those. Now, these kinds of polymorphisms are really rare for non-hybrid Solidago species. And so, again, this is providing good evidence that Houghton's goldenrod is of hybrid origin. Now, what can we predict from our nuclear DNA with regard to our question of who are the likely parental species, the progenitors of this plant? In allopolyploids, the parental nuclear genes are going to be duplicated. And the duplicated genes in the hybrid are referred to as homologs. Now, that's probably a term that only plant people understand, at least for the most part. And so our homologs that we'll be talking about here are those duplicated regions that are duplicated because they originated from different parents. And so if you were to have a tetraploid, a hybrid that was tetraploid, you would expect to have two divergent copies of that particular genetic region. If you have a hexaploid like Houghton's goldenrod, you'd expect to find 3 divergent regions. And an octoploid, like that Michigan disjunct population, you'd expect to find 4. And so we'll be able to use molecular cloning to separate those homologs from our hybrid, and then we should find that they are phylogenetically sister to their orthologs, the copies that we find in the parental species. So this is pretty busy because I used 26 different species of Solidago, and then we've got all the different clones from the 4 different geographical regions. So let me just point out a couple things, and then we'll focus up on the part of this phylogenetic tree that we're really interested in. And so first off, with very high support, 100% support, that the whole genus Solidago is monophyletic. There is something that was unexpected, however, and that is the fact that one of our flat-topped goldenrods that's in this taxonomic section, Tarmacoideae, is actually paraphyletic. It is not with the other flat-topped goldenrods in phylogenetic analysis. So we're going to focus up on this portion of the tree. That's what we're interested in here, our sequences received from cloning of Houghton's goldenrod. So I'll start down here at the bottom with Solidago ridellii, or what I've identified as the ridellii clade. So what we found is that the clone types from the northern specimen I collected from northern Michigan and the specimen from Canada are sister to Solidago ridellii, and a strong support for that relationship. This clone that was recovered from the disjunct population in Michigan, that one's actually a chimeric sequence. It shows evidence of ridellii, but it shows evidence of a second parent as well. So through biological recombination, we can get a chimeric sequence. And so that is a weak support for that relationship. But this group here shows strong evidence that Solidago ridellii is one of the parental species of Houghton's goldenrod. Now, the rest of my clones have resolved with other members of section Tarmacoideae. And so Solidago tarmacoides was a species that was presented or hypothesized to be one of the parentals. Well, when I sequence that, there's always things that come up that you're not expecting. When I sequence Solidago tarmacoides, its sequence was very messy. A messy sequence indicates that you likely have length mutations within that individual, indicating that there may be more than one ITS type within that individual. So I ended up having to sequence this as well. So I have a Solidago tarmacoides, a type 1, one of the 2 sequences recovered, and then a type 2. So in this subclade, 3 of my accessions, 3 different geographical areas, produced clones that resolved with Solidago tarmacoides type 1. The Michigan disjunct population also produced a clone that resolved with Solidago tarmacoides type 2. So these relationships here, subclades A and B, provide evidence that Solidago tarmacoides is a second parent of Houghton's goldenrod. Now, this subclade C, fairly low support, but I'll explain why. All of these 4 different accessions from 4 different geographical areas, they all had a unique base pair, just one. It was a unique cytosine in their ITS2. That perhaps indicates to us that this was a single origin for this species. But in phylogenetic analysis, you need more than just one character to get a strong support value for that relationship. So there is support. It is weak, but indicating likely that it is of single origin. And then likewise up here, these 2 clone types, this is a Solidago ohioensis. That sequence was obtained from GenBank, the national database of genes, and it had one unique base pair in the external transcribe spacer, and that was shared with Solidago rigida. And so again, there's a group here where these 2 clones are weakly supported, but we cannot indicate, this relationship doesn't indicate parentage because we've got 2 different species that are present there. So the remainder of my clone types are part of this unresolved, what we call a polytomy. In that polytomy is the proposed parent Solidago ohioensis. So through phylogenetic analysis, we were not able to determine who the 3rd parent was. There's not enough informative data to indicate ohioensis as a parent. So this is where statistical parsimony network analysis comes into play. This looks at things in a different way. Rather than in phylogenetics, you work from your hypothesized ancestors out to the derived species. This starts with what you have, the derived species, and works backwards in a coalescence. And so what network analysis does is it takes all those DNA sequences that are exactly alike, and it groups them together and puts a circle around them. So I've separated them so we can read them. So this network of my 26 Solidago species and the clone types is supportive of what we see in phylogenetic analysis. We have a Ridellii clade where clones from Houghton's are identical to the sequences of Solidago Ridellii. We have the Solidago tarmacoides as a parent. Both types 1 and 2 are forming groups with my clones. And this is then where phylogenetic analysis could not resolve that polytomy. Network analysis indicates that Houghton's from all 4 different geographical locations has an identical DNA sequence to Solidago ohioensis. This is our unique cytosine, the 1 base pair that they all shared. In network analysis, these single lines indicate 1 mutational step. So that unique cytosine is that 1 mutational step difference from ohioensis. GenBank ohioensis is 1 mutation different from my accession that I sampled in the field. And so these are likely all originated sometime in the past from that Solidago ohioensis. This indicates that that is a third parent present in the nuclear genome. So for a hexaploid, we're expecting to get potentially 3 divergent copies, and we have them for Houghton's. So now, with regards to my third question of is this a single hybridization event or did Houghton's form from multiple hybridization events? If it's single, we'd expect that all of our accessions are going to have the same parental homolog types. Versus multiple origins, we should see the different entities having their own unique homolog types. So, as we sort of sum up everything I was just talking about, a single origin, I believe, is strongly suggested for this species. Pringle's entity number 1, the primary distribution throughout northern Michigan, saw clear evidence for Solidago ridellii, the Tarmacoides type 1, and Solidago ohioensis. Entity 2 in Canada, clear evidence for ridellii and ohioensis, but we didn't recover Solidago tarmacoides. Now that doesn't mean it's not there or that it didn't contribute. It means that through cloning, that was not recovered. So one would need to sequence additional species, clone additional accessions, and that in an effort to find that. However, there are biological processes that occur frequently in hybrid plants called concerted evolution. And concerted evolution, through processes like unequal crossing over that you're probably familiar with, acts to actually homogenize the DNA. So in several well-known and well-researched plant species like cotton and sunflowers, we know that concerted evolution has actually eliminated sequences from one of the parents. Perhaps in the Canadian populations that are somewhat isolated from northern Michigan populations, that also could account for why we haven't found it. But Solidago tarmacoides morphology strongly suggests that it is a parental species. It is likely the only goldenrod that could have contributed the large ray flowers that we find in Houghton's goldenrod. So everyone has always suggested that is a parental. So likely it is, we just didn't recover it. The disjunct population in Michigan, we have evidence for ridellii. Again, that was a chimeric sequence, so it's not a complete match. But we have evidence for tarmacoides and ohioensis. This is our octoploid population. So we did find 4 types here, as we might expect with an octoploid. And then our New York disjunct population, again, ridellii is chimeric, so not completely identical, but again, evidence of its presence and then strong evidence for tarmacoides and ohioensis. So they all seem to be sharing a similar parentage. Of course, that's suggestive of a single origin. However, it is possible that it is still 4 different origins. It would just mean that these 3 parental species contributed in identical events in the 4 different geographical areas. And answering that kind of question can be difficult. It would require that we broadly sample all the parents, do a lot more cloning and sequencing to look for any variation that we might find geographically in these parental species. So more work would need to be done to thoroughly answer that question. All right, I also used chloroplast DNA in this project. And again, we're going to do PCR reactions. And I chose to use 4 separate chloroplast intergenic spacers. Now, intergenic spacers are non-coding, and so they're not under any evolutionary constraint. They're free to mutate and obtain differences that are useful in phylogenetic analysis. So using something that is not under any evolutionary constraint is important especially when you're looking at species that are very closely related. So we're looking within the genus Solidago, and then we're looking within an individual that's a hybrid. So you want to use something like an intergenic spacer. Now, for these intergenic spacers, they frequently have length mutations that are the result of indels, or insertions and deletions of DNA sequence. Those have been shown to be phylogenetically informative. And so I coded those indels and included them in my analysis. So what can we predict when we're using chloroplast DNA with regard to our question of who are the progenitor species? So since chloroplasts are maternally inherited in the angiosperms and in the Asteraceae, then we would expect that the sequences obtained from the chloroplast DNA would form a clade with the maternal parent. And I would expect then that that maternal parent is either going to be Ridellii, Tarmacoides, or Ohioensis, because those are the three that we found in the nucleus. So here's our first marker. This is a section of aligned sequence data in the intergenic spacer between the ACCD and PSAI exons. And here we find a unique 29 base pair indel that's shared by Houghton's from all 4 different geographic locations, again providing evidence from the chloroplast of a likely single origin. But notice we are expecting a member of section Tarmacoides, either Ohioensis, Solidago Tarmacoides, or Ridellii, to have that same indel, and they don't. So as I sequence through the other 26 species, all of a sudden, Solidago gigantea shows up with that same unique 29 base pair indel. Well, the first sequence for Solidago gigantea, you don't run out and tell your advisor that you found a match, because no one's expecting this plant to be a maternal source of Houghton's, and so immediately you say, oh, I have contamination somewhere. So you start from the beginning and you re-extract DNA, you do your PCRs again, you're very careful that you have no contamination, and oh, it's still there. Well, that must be real, because now I've been extremely careful, and we tell the advisor, and we sequence a few other accessions. Solidago gigantea from northern Michigan, these two, and from Canada have that same unique indel. This Solidago gigantea is from New York, and we don't see it there. And so there are reasons for that. The Solidago gigantea is actually has different numbers of chromosomes in different geographical regions. So that's another story, and we can talk about that later. So what are our other markers going to tell us? Are we going to see that same relationship as we look at the 3 other markers? Well, as we look at the PSBA and TERN-H marker, yes, we do see that. Here we have a 15 base pair deletion, again shared by Houghton's from all 4 different geographic locations, shared with gigantea from northern Michigan and Canada, but not from New York. We have 2 additional markers that I looked at. And so here we see a 5 base pair deletion, and we see microsatellite length variation shared by these. However, there are other species that have those same length mutations. So that does not refute our data, but based on this alone, we could not make any statement about that maternal relationship. One more marker that we looked at, and again, here we have microsatellite length mutations, but those same length mutations are shared with other species. Now, microsatellites are these strings of repeated data, repeated nucleotides, and they are well known to be homoplastic. They are shared not because of common ancestry, but because of processes like unequal crossing over, they're likely to occur. So they can be useful, but we have to be careful in phylogenetic analysis not to include homoplastic data. So here is a strict consensus tree of the chloroplast sequences from all of the Solidago species. Again, using chloroplast data, Solidago is a monophyletic group. Here we see a sister relationship between Houghton's and Solidago gigantea from northern Michigan and Canada. This topology of our tree, of course, is incongruent with the one I showed you earlier from the nuclear DNA. When you have an incongruence between chloroplast and nuclear, that's also used as evidence for a hybrid origin. So let's look at this a little bit more. How could we explain these data? We were expecting to find either Redellii, Tarmacoides, or Ohioensis as the maternal parent. However, we did not recover Solidago gigantea from the nuclear genome. We were expecting 3 species. We have a hexaploid, but one of them was not Solidago gigantea. I sequenced about 30 clones for each of those 4 accessions, so that's a lot of sequences. Didn't recover any gigantea. Then I went as far as designing specific primers that were only going to amplify gigantea, and tried to amplify it from Houghton's, still didn't find it. So we would not really want to say that there's a maternal relationship here. So how else could we explain the presence of Solidago gigantea chloroplast DNA in Houghton's goldenrod if it's not a maternal relationship? Well, again, these strange plants do lots of strange things. And one thing that they do, and many plants do this, is a process that's called chloroplast capture. Now, chloroplast capture is involving a hybridization event that is intergressive hybridization, meaning that the chloroplast of one species is moved into another species without the nuclear genome. This occurs through forming a hybrid. That hybrid acts as the maternal parent and repeatedly back crosses to the other parent. And so over time, and it can actually happen fairly quickly, the chloroplast is moving along, but the nuclear genome is erased through the repeated back crossings to the other parent. And so happens in many, many plants. Chloroplast capture is well documented. And so I believe that for this data, chloroplast capture is our most parsimonious explanation for how we get the chloroplast in Houghton's goldenrod. So this is the first part of the study, and the conclusions here, Houghton's appears to be of hybrid origin, as the polyploid nature of it would suggest. We seem to have good evidence from the nucleus of these 3 parents for the plant. Also have evidence from the chloroplast of Solidago gigantea, and again, arising through intergressive hybridization leading to chloroplast capture. And then suggested that it has formed only once. So with that, I will take a break here for a minute. And the second part of the presentation is on the population genetics for this species, so sort of a different angle altogether here. I think some of the more interesting stuff is there because it'll get into some of the glacial history of Michigan and that. But I think some of you were planning on about 45 minutes, which is about where we're at right now. And so I just thought I would be kind to all of you, and if any of you wish to exit, this would be a good time. And if not, that makes me feel really good. And shall we just continue on then? Or do you want to ask a few questions right now? Anything that really needs to get cleared up before we move on? Is that John? Yeah. Okay. I just wonder, how long did it take you to do all your research? Oh my goodness, we're going to get into the length of time. Well, I'll tell you. I'm a non-traditional student. And so it took me longer than a traditional student, but it took 10 years from start to finish. Of course, I worked full-time here and doing what I love, teaching you guys botany. And in addition, of course, as an adult with a family, there's all those responsibilities as well. So it took me a little longer than most would. Anything else that's pressing at this moment? All right, well, let's look at the population genetics then for those of you that are game for hanging on for a little bit longer. All right, so the second aim of this project then is to characterize the population genetic structure. And I'm going to do that again by using chloroplast DNA. So I sort of have four things I want to look at here. I want to assess how that diversity is structured within and among the populations. I'd like to estimate the relationships between genetic distance and then geographical distance. So are the individual subpopulations isolated from each other by distance, or are genes able to move? And since I'm using chloroplasts, we're really asking, are seeds able to move between one population area and another? Third, I want to look at habitat fragmentation. This is a shoreline species. Most of us have been up north, and we've seen some of the habitat fragmentation that's been occurring probably in the last 100 years or so, as we have cottages for recreation, home sites, beaches with parking lots and things like that. So has that habitat fragmentation affected the genetic diversity of this species? And then lastly, you can assess the potential origin, the phylogenetic origin of the species, and that's some of what I think is kind of pretty fun stuff. So again, I'll just briefly do methods here. I sampled 23 of the 70 known populations from across the geographic range. Again, a non-destructive sampling. This is a threatened species. So out in the field, I collected leaves from 60 random individuals. And then when you took that back in the lab, I used 20 of those in the genetic analysis. Again, I'm using the chloroplast markers that I used before, because I know from the previous study that there's going to be some length variation there. And I'm doing a method called fragment analysis, and it's performed through automated capillary electrophoresis. So when you're doing your PCR, you add to the end of your PCR product just a single fluorescent dye. That goes through the capillary. The automated process reads that fluorescent dye and tells you this fragment length is 450 base pairs, this fragment length is 429, etc. And then I did that for the 3 different markers and combined them to form haplotypes for the different individuals. So why should we end up using chloroplast DNA in a study such as this? Well, it is non-recombinant and it is maternally inherited. So that's going to allow us then to look at both the historical and the current contribution of seed movement to the overall genetic structure of the population. And that's important because seeds are acting both as the migrants and as the founders of new populations. And of course, for a threatened species, establishing new populations would be a goal that you would want to have in mind. Also, the genome is haploid. Now, a haploid genome gives you what we call a smaller effective population size. That's going to accelerate the process of genetic drift. So you're going to be able to see differentiation between the subpopulations sooner than you would if you used nuclear genes. Nuclear genes have contributions from 2 parents, and so it takes a lot longer to see the effects of drift and of the fragmentation. So chloroplast was the way I wanted to go because habitat fragmentations probably only occurred in the last 100 years or so, and I wanted to assess that. So here's the map I put together. Where the little pie graphs are showing not only the locations that I collected from, but we also have then the 14 different haplotypes that were recovered and the distribution of those haplotypes. A couple things to point out before we really talk a little bit more about each of the aims. One I wanted you to see is that here in the lower peninsula, almost all of our populations are monomorphic. There's only one haplotype there. We don't have the diversity of haplotypes that we see in some of these other populations. Second thing I wanted to point out will be bringing this back up again in a little bit, there's a couple of pretty interesting sites for Houghton's goldenrod. One is up here at what is called Summerby Swamp. But Summerby Swamp is actually a marl fen. You have to be very careful there because you can sink in the marl like if it was quicksand. That's a marl fen, and it is very similar to the New York population, which is also a marl fen. Now, most of these other populations are shoreline, the interdunal hollows in that along the shores of Lakes Michigan. And so we'll bring that back up here in a minute as we work our way through. So, first thing I calculated was a measure of gene diversity. This is calculated as the sum of the squares of the different haplotypes. And the gene diversities range from 0, there is no diversity, to 1, which indicates that the haplotypes are evenly distributed across all of the sampled populations. Now, unexpectedly and pleasingly, Houghton's goldenrod had a fairly high 0.76 overall gene diversity. So across the entire population, that's good. Most rare species, like these two that can be found in Michigan as well, have a very low gene diversity. So that's a good thing. However, when I looked at just the within a subpopulation, well, we have a bunch, we have 11 out of 23 that were monomorphic. There's no gene diversity there at all, right? And so if you take the average of these 23, the average is very low still, and probably more in line what we would expect for a rare species. So we'll look at that and why that might be here in just a moment. Now, I wanted to test population structure, and that is done with a statistical method called F-statistics that basically looks at the degree of differentiation between those subpopulations. You test it against the null hypothesis, so an FST of 0 indicates no variation between our subpopulations, and a 1, all the variation is between the subpopulations. So what can we predict about population structure? So if Solidago Houghtonii is just one large population, seeds are moving across the entire geographical area, then we would expect our FST value to be near 0. And if it has restricted gene flow and seeds aren't moving between those different populations, then our FST value would be close to 1, indicating that most of the variation is occurring between the different subpopulations. So when we did that analysis across the 23 different populations, we did get a fairly high FST value as well. So we do have significant structuring in these populations and a significant deviation from that null hypothesis. Now, isolation by distance. What are we going to be able to predict here with this idea of isolation by distance? Can seeds move between the different populations? So if the subpopulations are isolated by distance, what we expect is that those populations that are really close together are going to be more genetically alike because there's a better chance that seeds can move to another population that's very close. And that they will be more genetically different when they're separated by quite a distance. And so we're going to be interested here then in our seed ability to move. And so we will be looking at things like, is wind effective at dispersing these seeds over long distances or just in short distances? And that'll affect our spatial structuring. Also did some statistical methods with that. Basically, the FST value can be used to calculate a measure of gene flow. And then a mantle test compares two matrices, one of pairwise genetic distance with a matrix of pairwise geographical distances. A mantle test, these matrices are not necessarily independent of each other, right? And so a mantle test is used to compare these matrices that do have some relationship between them. So we're going to be looking at our R value, and a positive R value will indicate that we have isolation by distance. So when I did all 23 populations, I did get a slight positive indicating that there's some correlation between genetic distance and geographical distance, but at a 95% confidence interval, this was not significant. And so that was kind of curious to me, as I've spent a lot of time out there with Houghton's goldenrod. It seemed perfectly good habitat, but it's not being colonized. It appears to be isolated by distance, and so I wasn't sure about what was up with that. So I looked at the individual FST values and found that the FST values for that New York population were very, very similar with the summer bee swamp, those two marl fens. Now, what is known is that when there is a long distance dispersal event, so a seed somehow perhaps moving from the Upper Peninsula in Michigan over into New York, these long distance dispersal events act to homogenize the population at a landscape level. And in this kind of work, long distance is considered to be 1 kilometer or more. New York's over 600 kilometers away from this northern Michigan population. And so that appeared to be homogenizing my data and giving me not a significant isolation by distance. So I removed the New York population from the data set, got rid of that long distance gene dispersal event. And sure enough, our value is more significant, highly significant here. So our populations are isolated from each other by distance. Seeds are not likely moving very far. These are goldenrod seeds, and they have what's called a pappus at the top, which helps in wind dispersal. You can see that they're quite small and they're fairly light. One might think then with this structure they'd be pretty good at flying and moving around, but actually our calculated number for gene flow is fairly low. Other people have studied seed movement in goldenrods, other species than the one I'm looking at, and found that on average they're only moving about 15 meters. And so along the shoreline of Lake Michigan, appropriate habitat for Solidago houghtonii is spread out more than just 15 meters. Of course, some of them move in winter over some of the frozen land. They can skate along a little bit better. A few species are known to sort of move along water, and this hasn't been looked at for Houghton's. Water might be an option for it because it is a lakeshore species. But yet it does seem to be isolated by distance. Seeds don't appear to be moving very far. Now, next question I wanted to look at is what's the effect of habitat fragmentation? And so our fragmented, or populations that are fragmented by human activities, are they going to be more genetically isolated than populations that are less disturbed? And so here's a typical scene. This is the Mackinac City beach. This area here is prime Houghton goldenrod habitat. Here's a small foredune. It's an interdunal hollow. It gets a little damp and wet in there. Perfect place for it to grow. And indeed, it was growing there. But one would think that this was continuous. Well, it got turned into a parking lot. This is down as you head down towards the little town of Cecil. These are shoreline homes. You can see the property here. They left it near the road, undisturbed. Houghton's is growing there, but their neighbor decided that up in northern Michigan, it would look great to have a lawn, right? So they got rid of Houghton's habitat. And so that is a typical scene. So the population has been fragmented. And indeed then, our regional lower Michigan FST value is near 1. So indicating that we have nearly complete differentiation between those subpopulations. If we cross the water and we go to one of my favorite sites, you love to camp, this is a great place to go. It's a state forest called Big Knob, and this area has perhaps one of the largest Houghton goldenrod populations. Perhaps close to 4 miles of beachfront is covered with this plant. And so We see nice sort of interdunal wetland area here, and in this narrow little path, we find Houghton's goldenrod. But that's a similar scene up and down the beach in the Upper Peninsula, where we haven't had as much human influence on that habitat. And so our FST value, while it's still high and populations are still subdivided, it is lower than we found in the Lower Peninsula. So we still have about 27% of the variation within each of those subpopulations, and most of them were not monomorphic as they were in the Lower Peninsula. So the last question that we can deal with here with population genetics is, what is the phylogenetic origin for Solidago houghtonii? And here we're going to be looking for patterns of congruence between the genealogical relationships of our haplotypes and then their geographical distribution. And through that, we can predict where this plant originated. And so this is pretty busy, and I know that, but basically I want to focus here. This is all work done from a paper by Schall in 1998 dealing with phylogeography. Very good image as a starter to begin to explain what is going on here. So we're going to do statistical parsimony network analysis. And this is the genealogy of the haplotypes, her haplotypes A through G. And what can be predicted from this is that the ancestral haplotypes would be centrally located in your genealogy, so C and G in the example here. And what we'd expect to see when we look at that geographically is that those should be very widespread. And so we're looking for that congruence between geographical distribution and the genealogical network. And so here's the geographical distribution when we do see congruence. So the haplotypes labeled C and G are quite widespread. The derived haplotypes located at the tips in the network are isolated from that main distribution. That's congruence. We're looking to see that. That would indicate that the haplotypes, C and G, are ancestral. We hope to see a similar pattern with Houghton's. Now, C here is incongruent. We've got C and G with our haplotypes that occur out at the tips, and they're scattered throughout the geographical distribution. This type of arrangement is thought to result when there's ancestral polymorphisms. The population as a whole is polymorphic for its haplotypes. Lastly, we generally have congruence except when it comes to E. E is out here, derived position, but then E shows up in here as well. That type of pattern is suggestive of a long-distance gene dispersal event. So what do we see when it comes to my data? Here's our map again, and here is my statistical parsimony network analysis, and haplotype 7 is centrally located in the network. Haplotype 7 is widespread. Some of it here, we see the green, by the way, I'll just point that out, green here, haplotype 7, throughout Upper Lower Peninsula, throughout the Canadian distributions, but not in New York. So we generally see congruence here, and suggestive that haplotype 7, that this is the ancestral haplotype for this species. Haplotypes 14 and 3 are definitely in derived positions, and it appears that haplotype 3 is derived from 14. Well, where are we going to find those? 14, the fuchsia, and 3 here, the orange. So haplotype 14 is located here in that marl fen at Summerby Swamp. Haplotype 3 derived from it is located over here in New York. 9 is also out at a tip, derived. 9, so these two do share an awful lot of similarities. That is not congruent, by the way. New York, these similarities, you should find that in a population that's very close, right? So that's an incongruence suggestive of that long-distance gene dispersal event that we looked at when we talked about isolation by distance. There's one other place on this network where we also see incongruence, and that is here on the Manitoulin Island. Haplotype number 4 also shows up in our Michigan disjunct population. Again, that could be ancestral polymorphism, it could be a long-distance gene dispersal event. But in general, the majority of this, we do see congruence between our network and our geographical distribution. So this is suggestive that Houghton's goldenrod originated in northern Michigan, likely somewhere around the central distribution of haplotype 7. So if it originated here in northern Michigan, how could it have spread across all of this open water? Well, here we have to look at our history of the Great Lakes area. All of this area where Houghton's is located was glaciated until about 10,000 years ago. That was when we had our last glacial maxima. Following the retreat of the glaciers, lake levels rose for a little while, but then about 6,000 years ago, at a time period that we refer to as the hypsothermal, environment was warmer than it is today, and lake levels in the Great Lakes lowered significantly lower than they are today, so that all of these islands were connected, and this was all connected to the Bruce Peninsula. This was all dry land. We have great evidence of that because down here in the Georgian Bay, there are trees that are rooted to the base that, of course, now are dead. They're underwater, but how did they get rooted to the base? Well, it was once dry land, right? And so it seems that perhaps during that warmer time period, there may have been a range expansion traveling along that Niagara escarpment that was all connected. Lake levels, the climate cooled, lake levels again began to rise again. They reached their current level about 3,000 years ago. And of course, rising lake levels then broke these landmass into the various islands in that of the Manitoulin region, making it much more difficult for genes to move between these areas, leaving isolated populations that then were free to begin to accumulate their own genetic mutations. And we see then closely related haplotypes in similar geographical areas. So, in conclusion of this part, Houghton's goldenrod appears to have a northern Michigan origin. We seem to have fairly high genetic diversity across the whole entire species, indicating that we likely should protect all of the habitats throughout Canada and northern Michigan in that if we want to maintain that higher level of genetic diversity. And then protecting some of those less disturbed habitats like the Upper Peninsula would be a good start to maintaining genetic diversity for this species. So there was lots of people to thank, lots of individuals that allowed me to collect on their property, and as well as individuals that helped to fund this project. DNA research is always quite expensive, and so I received a grant from the DNR that the citizens of the state helped to fund. Haynes Foundation and Michigan Botanical Foundation. Those of you that are going to be going off to grad school someday, there's a whole list of people that have worked in the lab. Everyone helps each other, and that's the idea of cooperation in the lab, and then, of course, my committee members. And so even GRCC needs to be thanked for allowing me freedom to work and teach and go to school at the same time. So that's where I'm finished. And so are there any additional questions that you guys have? Wow, you either went like this or I just answered it all.
I have one really simple question.
Yes.
If you showed the disjunct populations after the last glacial period,
Mm-hmm.
Is it possible that you said you find locations where you think you should find Houghton's goldenrod, but you don't?
Right, these nice interdunal hollows, right?
Right. And so I always have the same problem too. I always wonder, why isn't that insect there? What did you come up with? What is it?
Well, it looks like seeds, at least, are not dispersing long enough distances to make it to those locations. Is there a possibility that one could transplant things and see if they'd take in those habitats? That's always a possibility, but when you start having unique genetic makeups in the isolated populations, usually that kind of movement doesn't often work. So I think it's just the fact that gene dispersal is so low. Now, we were just looking at seed movement, right? We haven't considered nuclear genes yet, and so we might have a different story if we looked at nuclear genes, but we chose chloroplasts because we wanted to see evidence of recent events. So, any other questions? Tim?
Yeah, does the plant have any particular ecological importance or like an economic importance?
Yeah, right. Why should we save this thing, right? That's what you're asking, right? Why should we save these things? There are some species of goldenrod that some people claim have some medicinal benefits. I don't think this one has been looked at. Mostly the weedy varieties have. I think its biggest benefit is creating a gorgeous habitat for man to enjoy. Without this species and others that are like it along the shorelines, it just wouldn't be the same place for any of us to go to visit. So, no, but I do believe that there are intrinsic value to each and every species, and maybe not an economic value to this one. That answer your question?
Yeah, or the other part was the ecological value too, I guess. For anything or food supply for anything.
Well, definitely, it provides nectar to numerous generalist pollinators. So there's 30, 40 different things that you can find, you'll have to identify them, that are visiting this plant and feeding on the nectar. And so it is a food source for the smaller insects and that. It is, a lot of them lay their eggs on it, and you'll find some of their larvae and that crawling around in amongst the inflorescence. They'll eventually chew on some of those seeds, but that's an advantage of having lots of seeds in one inflorescence. So in that point of view, yes, there's an ecological value to that species.
What prompted you to choose this particular species?
What prompted me to choose this one? That's actually a really good question. That's a fun one for me to answer. I always want to talk about that, but don't think it should. Back when I was an undergrad, I took a summer course at the University of Michigan Biological Station called Boreal Flora. And so this is the northern Michigan boreal flora. So I was introduced to the plant. And the instructor indicated there was a lot of questions that we just don't know. Is this a hybrid? Where'd it come from? And also this geographical distribution with the Niagara Escarpment and all that, it just piqued my interest. And so it was basically an undergrad instructor. And as I got into grad school, it's a Michigan plant. I'm a Michigan girl, so it just fit, so I chose it.
When you were out collecting, what criteria did you use to say this is Houghton's goldenrod and not
Right.
some other one?
Right.
Because that could highly contaminate.
Sure, definitely would.
Got the wrong species.
And the one in particular, Ohioensis, very morphologically similar if you stand back from it. Of course, once you start studying goldenrods, you can pick up all those little differences. So Houghton's, because it's a hybrid, is the inflorescence, the individual florets are going to be much larger than they are in Houghton's. The numbers of them are fewer. They're bigger and fewer numbers. Ohioensis can have 400 florets in its inflorescence. But the big thing you want to check is Houghton's has hairs in the inflorescence, and Ohioensis does not. So you got to get your hand lens out, you got to look for hairs on the plant, and then that will help to guarantee that. You certainly would not collect in a vegetative state. They are too similar from just the green material. You have to have it in inflorescence. They're also clonal species. I didn't mention that. They form little clumps that are clones. So I did not collect, there were several meters between one collection and another. The clones tend to stay relatively close together because that would have also skewed the data results.
What time of the year do you have, and what window do you
Yeah,
have?
small window.
And then how did you identify the New York population?
Okay, so let's see, what was the first part of that question again?
What time of the year?
So August, and so it was always, that was hard because that's backing right up to when school starts. And so it's a protected species, so it does require you have to go through all the permitting to do that. So you really need to start thinking about your collection 6 months ahead of when you're going to go do it. You have to, I had state, federal permits, permission from the Native American tribes. They require that you bring an offering for each plant that you sample. You have to give an offering back to Mother Earth. That's tobacco. And so I had to learn all these cultural things as well, so that was kind of cool. And so that was basically this late summer kind of collection. And so, you know, zooming around the lake, I camped all along the way. My husband's not a camper, so I brought other friends with. Now, about the New York population. That's been documented to be there for quite a while. In the 1930s, a group called the Burgeon Swamp Preservation Society formed to protect this unique area, the Burgeon Swamp. They actually have an extremely high number of endangered species there. And in New York, that's the only place it grows, and we're talking just a few acres. So it's an endangered plant in New York. And so that was documented, and others that have talked about that. So it was kind of a known thing that it's in New York. But again, getting permission from them, one of their big problems was if we give you permission to collect this plant, and you find out that indeed it is a different species, then we have lost our protected status for our plant. And we would have to start all over again obtaining that. That was a huge concern for them. But they came out on the side of science, that this would be good to know no matter what they have to go through to get it reprotected. But again, it looks like it is all one species. So they're happy. These are great questions. Anything else? By the way, I should just mention to all of you, Robert Riggio is here, and Robert did an honors project with me last semester, and he kind of kept me motivated on looking at chromosome counts for Houghton's goldenrod and worked some with me on that project. That's part of the project's not finished yet. Tried like crazy to get it done for the dissertation, but didn't. Partly because it looks like I'm finding both octoploids and hexaploids in this disjunct population, and all that needs to get sorted out, so that's a whole other mess. All right. Well, I guess it's time to go get some food. And thank you all for putting up with this long talk.